Hi, I'm Grace Davis, and this is my video assignment. In eukaryotic cells, there are two major phases of cell cycle stages, known as interphase and the mitotic phase. During interphase, the cell grows and makes a copy of its DNA. The mitotic phase is the time of division when the cell separates its DNA into two sets and divides its cytoplasm. This results in forming two new cells. During interphase, the cell prepares for division and can be further divided into three phases, G1, S, and G2. Here is a photo of two cells in interphase. During the G1 phase, the cell grows physically larger, duplicates organelles, and creates the molecular building blocks necessary for the cell to complete later steps. During the S phase, the cell synthesizes a new complete copy of DNA inside the cell nucleus. Furthermore, centrosomes are duplicated to later help with separating DNA during the mitotic phase. Finally, during the G2 phase, the cell grows even more while creating proteins and organelles. It also begins to reorganize and compartmentalize its contents in preparation for mitosis. The G2 phase ends when mitosis begins. Mitosis occurs in four different phases called prophase, prometaphase, metaphase, anaphase, and telophase. And here is a little chart that I found going through all the steps, and I will be going through them in more detail. During prophase, chromatin condenses into chromosomes, the nucleolus breaks down, and the mitotic spindle begins to form. In animal cells, the centrioles near the nucleus begin to separate and move to opposite sides of the cells. Kinetochores on the spindle attach to the centromeres of sister chromatids. Then, prometaphase occurs between prophase and metaphase and consists of the nuclear membrane breaking down and chromosomes condensing. Next, there is metaphase, where the spindle fibers attach to the centromere of each pair of sister chromatids. The sister chromatids line up at the equator of the cell at the metaphase plate, which is sort of in the middle of the cell. The spindle fibers help separate the sister chromatids and ensure that they are allocated to different daughter cells during cell division. Next, during anaphase, the kinetochore microtubules pull chromosomes towards the poles of the cells, and the microtubules push the poles apart. And finally, there is telophase, where the spindle disappears, the nuclear membrane reforms, the nucleolus reappears, and the chromosomes start to decondense. This prepares the cell for division. And I found this nice photo of all the different phases, prophase, metaphase, anaphase, and telophase. It left out prophase or prometaphase, which is in between prophase and metaphase, because some people don't include that in all of the steps, but I decided to. And I just thought this was really pretty and really shows what is going on during all of the phases. Cytokinesis is the division of the cytoplasm and the cell contents, but not the actual chromosomal DNA. Cytokinesis is when the division of the cytoplasm forms two new cells, and cytokinesis sort of overlaps with the final stages of mitosis. It may start in either anaphase or telophase. It depends on the cell, and it finishes shortly after telophase. There are differences between animal cells and plant cells when it comes to cytokinesis, and I found another cool chart here, and I'll have all of the references to these photos linked. The differences are really based on the structure of animal and plant cells. As you know, animal cells are less rigid because they don't have a cell wall like plant cells. In animal cells, cytokinesis is contractile, which appears as pinching of the cell. There's a band of actin protein filaments and a cleavage furrow crease. Plant cells have a more rigid cell wall, so they do not divide in this manner. In contrast, plant cells have a structure called the cell plate that forms down the center of the cell. The cell plate effectively splits into two daughter cells separated by a new wall. Thank you.